In this study, the authors aim to investigate how changes in RAS-ERK signaling dynamics affect gene expression and cell proliferation decisions. This is Figure 4A. In the experiment setup, they applied constant and post-input signal to both of the wild-type NIH33 with or without drug SB590885, which is a paradoxical BRAF inhibitor that slows signal deactivation by promoting BRAF dimerization. Cells were seeded in 384 wild plates, RAM-starved, and stimulated with either constant post-input signals. The stimulus patterns varied in on and off intervals to mimic diverse dynamic signaling scenarios. After stimulation, cells were fixed and immune-stained to detect expression of three downstream ERK targets, uh, ERK target expression targets, which are c-Jun, EGR1, and circulating D1. These gene expressions are the keys in controlling cell proliferation. For the figure 4b, in the first chart, we observe that constant RAF input leads to graduate increase in c-Jun expression in untreated cells. Means it has no drug treated. Under post-input, c-Jun expression remains low in wild-type NIH33 cells. However, in cells treated with drug SB590885, post-input leads to elevated c-Jun expression, comparable to that induced by constant stimulation. This suggests that drug SB590885 lowers the response threshold of c-Jun to transit RAS-ERK inputs, making cells more sensitive to weak or pulse signals. For the chart 2 EGR1 expression, continuous input in untreated cells leads to an initial peak in EGR1 expression, followed by an adaptive decrease, likely due to negative feedback regulation. Interestingly, post-input with drug SB590885 results in similar EGR1 expression pattern as constant input without the drug. This indicates that the drug-treated cell interprets post-input as if it were sustained, altering temporal decoding of the signal. In the third chart, circling D1 expression, so the stimulation pattern is slightly changed. So the on is the it is also 30 minutes interval to turning the stimulation on, but the 30 minutes off changed to 10 minutes off. Under the condition, circulating D1 expression increases over time, both in cell receiving constant input without the drug and cell receiving post-input with the drug. In contrast, post-input without the drug fails to significantly induce circulating D1 expression. Again, the presence of the drug SB590885 allows transit inputs to drive gene expression normally, only seen with sustained signals. In normal NIH33 cells, c-Jun and circulating D1 are induced by sustained input. EGR1 shows a peak and adaptation pattern under continuous input, but remain elevated under pulsed input when signaling deactivation is slowed. likely because short off intervals reduce the buildup of negative feedback. This data indicate that altering RAS-ERK signal kinetics through drug treatment shifts the filtering behavior of the pathway, making short subthreshold signals sufficient to activate proliferation-related genes. In summary, slowed RAS-ERK transmission dynamics also dynamics allow cells to misinterpret pulsed signals as sustained, leading to aberrant expression of key genes like c-Jun, EGR1, and circulating D1. This highlights how dynamic signal processing, not just amplitude, is critical for maintaining proper cellular responses, especially in pathways that regulate proliferation. Mm, Um, Next one. Drug-induced slowing of RAS-ERK kinetics expanded the growth regimen, enabling proliferation under normally non-proliferative inputs. Uh, for the experiment set up for the 4C, since the RAS-ERK pathway is a key driver of cell cycle entry, the earlier results showed that cyclin D1 expression is altered under slowed signaling condition. The author wanted to know, can delayed ERK deactivation lower the threshold for triggering proliferation? To test this, these stimulated NH3T3 cells with various dynamic RAS inputs for 19 hours in both the presence and absence of the BRAF inhibitor, which is the drug SB590885. Um, so the drug SB590885 is known to slow the decay of PP-ERK signaling. They then measured S-phase entry using EDU incorporation, a method to detect DNA synthesis. Each microvial received either no input, constant input, or pulse input with different on and off durations. For the heat map interpretation, the results are presented in the heat map in Figure 4C, where the x-axis here represents the on duration, like how long the signal stays active. The y-axis represents the off duration, like how long the signal is off. And the color intensity represents percentage of pro proliferating cells, like EDU plus cells. Control group is no drug. In wild type cells without drug, strong proliferation only occurred when on interval was long and the off interval was short. The pattern defines the so-called growth regimen, a narrow window of sustained input that capable of pushing cells into S shape. Signals that were too brief or had long off intervals were filtered out by the RAS-ERK pathways and did not trigger proliferation. With drug SB590885, expanded sensitivity. Now compare this to the heat map on the right with drug SB590885 treatment. We see a dramatic expansion of the growth regimen. Cells now proliferate in response to many input patterns that were previously non-proliferative. Even signals with shorter on times and longer off intervals are now interpreted as strong enough to drive the cell cycles. This confirms that no RAS-ERK signal deactivation effectively lowers the threshold for the proliferation. Figure 4c demonstrates that not only signal strength, but signal duration and kinetics are critical for determining whether cells into the cell cycle, the drug SB590885 treated cells become hypersensitive to dynamic signals, responding to inputs and normal cells would simply ignore. This has major implications for disease, especially cancer, where mutations of drugs that alter signaling dynamics may lead to inappropriate proliferation under normal physiological physio- physiological conditions. Um, Figure 40 is pretty straightforward. Long input signal and altered dynamic pathway, both causing proliferation. So the figure in the left described how constant input signal or long input signal stimulate on the cell will cause cell proliferation. However, the post or short input signal will not cause cell proliferation. It showed that Rosberg signal dynamics can reshape the cell division on proliferation. Under normal signaling condition, like the one on the left still, the Rosberg pathway acts as a temporal filter. The only strong and sustained input pass through and trigger cell proliferation. Post and weak signals are filtered out, maintaining proper control of cell fate. Uh, in contrast, in the diagram in the right shows what happens when rather kinetics are slowed by mutation or drug treatment. The pathway misinterprets short or weak signals as sustained. As a result, cell into the cell cycle inappropriately, even in response to input patterns that are normally non-proliferative. In the first two, the input signals in the right is like short, it's post, but the middle one can still cause proliferation here. So this overall misinterpretation of signal dynamics may explain how cancer mutations or paradoxical drugs can drive hyperproliferation.